Hello, everybody. Welcome to the BioRevolution lecture number 3, if I'm not mistaken. My name is Rüdiger Hell. I'm the Dean of the Faculty of Biosciences. And to be the Dean is always an honor. But there are special days where it is not only an honor, it is also a pleasure. This is a day with great pleasure for 3 reasons. The first one is I can welcome you to the #1 ranked biosciences faculty in Germany. So you choose the best spot in general and tonight and being here. And just to mention this, worldwide this is ranking number 32. I mean, we're up to Harvard and Cambridge, not so bad. The second reason is last Thursday, Heidelberg University was awarded 3 excellence clusters, and I'm very happy to tell you that biosciences are the only faculty that contributes to all 3 of these clusters. Yay! There are a lot of people involved. I just mentioned sort of the head representatives in 3D Matter Made to Order. It's Jochen Wittbrod, the former speaker, and still member, of course. There is SynthImmune, a combined medical synthetic biology cluster, where Kerstin Göpfrich, our colleague, is a co-speaker. And there is, of course, Green Robust, where Thomas Greb is a co-speaker, and where all the green people are, green background here, are involved. So this is really excellent for science. It means millions of euro pouring in every year on top of what we have. And of course, it means that this allows fostering junior researchers, and it will mirror back on our teaching. And if it had needed any evidence or additional evidence that biosciences is really the most innovative and interdisciplinary subject at this university, it is this contribution to the 3 clusters. I think there is a slogan from a bank in advertisement, and I would rephrase this: this central hub in life sciences is more than a faculty. The third reason why this is a pleasure tonight is the opportunity to introduce young faculty to the faculty, to have the opportunity to listen what they're up to, and to get a glimpse into the future of their work and the future work being done at the faculty for our students and probably the next excellence initiative. And with this being said, I would like to hand over to the Pro-Dean for Research at the Faculty of Biosciences, Professor Alexis Merzel, and he will guide us through the program tonight.
Thank you very much, Rüdiger. Yeah, welcome also from my side. I'm very happy to be here for the 3rd time and be able to welcome you to this BioRevolution event. So for the one that don't know exactly what that is, the idea of this event is that newly appointed faculty member here, those are the faces in the title, not only present their visionary research, that's the new idea in the title, but also emphasize how the importance of basic research is toward addressing societal challenges. And that's the new horizon in the title. So today we have Lauren Saunders and Gilles Storelli that's going to present. And both of them will give a short presentation, about 25 minutes or so. And this will be followed by a dialogue with you and that I will moderate. And you'll have the opportunity to ask questions, obviously. I'm just asking you to hold on to your question till the end of the second talk, and then we'll engage into the discussion at that moment, okay? So I think we could get started, and let me introduce Lauren first. So Lauren Saunders, she conducts research in developmental genomics, and she's exploring how cells' developmental history influence their function, and she used the developing fish embryo as a model for this. And her work basically aimed at uncovering the principle of vertebrate evolution and adaptation. So Lauren is originally from Chicago, and she did an undergrad in Minnesota, a very cold place. And before, that was before earning a PhD. And a PhD she actually earned in molecular and cell biology at the University of Washington on the West Coast in Seattle, where she investigated the hormonal control of the pigment cell lineage in zebrafish. And after completing a doctorate, that was in 2019, so not so long ago, she stayed in Seattle for a postdoc, as a postdoc for a short while, where she created the first detailed map of how every cell type developed in the zebrafish embryos, including those with specific gene modification. And she tracked what happened from these cells over time with very high resolution. And this offered a powerful new view on how genes shape development and why embryos that have the same genome can still very likely vary with one another. And this was actually a premiere in the field. So in 2023, she joined the DKFZ, so she came to Heidelberg here as a project leader. And she caught our attention, and maybe caught also our attention. And later on, about a year later, she was appointed here at COS as a tenure-track professor and Emmy Noether fellow to study the organismal and environmental adaptation of development here. So I'm very happy to have Lauren to present today, so join me in welcoming her on stage.
Hello, everyone. Can you all hear me okay, especially those in the back? Good? Excellent. Okay, thanks, Alexi, for that really nice introduction. I want to talk about the future today, but I think a lot of us know, especially here at COS who study evolution, that the future is really exciting, especially in light of what we know and learn about the past. So instead of telling you about the most recent research that we've been doing, which maybe you've seen at other talks recently, I'm actually going to go back into my research history and tell you a couple of stories that led me to where I am today and ultimately shape what we want to do in the future. And that theme is really about decoding diversity in biology and how we use multi-scale approaches to understand morphological evolution. And if you look around you in nature, you probably notice, as probably biologists here, that there is seemingly limitless diversity. And what you also know if you study biology is that this diversity arises from very small changes in DNA that happen over a very, very long period of time. And one of the fundamental challenges in developmental biology is to understand how these tiny changes in our DNA can translate across biological scales. And it's something that people have been trying to work out in development for a very long time. And it's been traditionally difficult because these scales are vast, right? We're talking about changes in genes and our chromosomes to cells that pattern tissues and organisms and ultimately up to populations and how these might change in different environments. Now, we are in an age where new technologies are revolutionizing developmental biology, and I think this is a really exciting place to be if you study this in this discipline. And of course, what we're doing now is building on foundational tools in developmental biology, which you might think of as forward genetic screens, animal transgenesis, whole genome and RNA sequencing, and advances in microscopy. But since about the 2010s, we've had sort of parallel revolutions in advanced live imaging, CRISPR genome editing, and single-cell omics that have really sort of launched this modern developmental biology area. And I think of this as kind of the integration point, which sort of spans these really rapidly advancing parallel timelines, and of course now is combined with advances in AI, multi-omics, and more sequenced genomes from different diverse species. With ultimately the goal being to connect molecular changes to morphological outcomes. So I think we're getting closer. There's still a ton of work to be done. And to explain how some of these integration of different methods work in practice, I'll share just a few examples from my scientific journey, and then ultimately how we're building on this in my lab to create scalable frameworks that will hopefully enable predictions of how organismal outcomes can arise from genetic variation. And in the bigger picture, I think that understanding these different connections has really important implications for conservation and evolutionary biology, regenerative medicine, and synthetic biology, which are also things that many of my colleagues in the faculty are interested in building on. So first, I'll talk about hormonal control of pigment patterns, which I worked on in my PhD. Then I'll talk about genetic control of scale formation in fish and why this might be interesting to any of us. And then finally, I'll talk about some of the new scalable analysis tools that we're using to study genetic perturbations. So many of you may know that hormones can coordinate post-embryonic development, and one of these is thyroid hormone, which controls the really massive changes in animals as they go from a larval to an adult stage. This is most prominent in animals like frogs and salamanders, where they have really drastic transitions and metamorphoses. And during my PhD, I used zebrafish as a model to study how thyroid hormone shapes the post-embryonic body plan. And one of the reasons that studying zebrafish was great for this over maybe frogs, for instance, is because while thyroid hormone is really important for post-embryonic development, zebrafish can still go through this process without it. So it gave us sort of a handle to understand this process. And in particular, I looked at one key aspect of post-embryonic development, which you can kind of see here, moving as a fuzzy striped pigment pattern in what I call a teenager zebrafish, to the mature adult pattern, which are the stripes, the namesake stripes of this animal. And the cells that give rise to this pattern all come from the same origin. They're also all neural crest derived. And zebrafish have 3 different pigment cell types, whereas mammals now just have 1. Zebrafish have black melanophores, iridescent iridophores, and yellow xanthophores, which you can see in the zoomed up version of the stripe, where you can see each cell is sort of contained, and you can see the individual colors. And using the powerful genetic tools that we have in zebrafish, I was able to remove thyroid hormone signaling by ablating the thyroid gland in the embryonic fish. And when I did this and looked at fish at about 3 weeks of age, you can see these, some differences in the pigment pattern, which are much more apparent when you zoom in. So in the stripe, as it's just becoming mature, usually you have the melanophores and the inner stripe of the xanthophores and iridophores, and then another stripe of black melanophores. The pigment cells here are contracted so that we can count them really easily. And without thyroid hormone, you have the iridophores there, and the xanthophores don't really seem to be there, but they're filled in instead by these black melanophores. So there we see these sort of cellular phenotypes. And a colleague before I joined the lab made these really beautiful movies over pattern formation to understand how these pattern differences arise at the cellular level. But we were still missing information about the molecules and the gene expression that was sort of changing in response to this. And this was a key question because thyroid hormone, which you may not know, I know Gilles does, is a hormone that binds to nuclear receptors. And nuclear receptors are these really amazing proteins that are both receptors and transcription factors. And so the thyroid hormone receptors were, once they were bound, they were going and changing lots of gene expression, and I wanted to understand what those changes were. So I asked, how do the changes in thyroid hormone translate into the cellular and organismal phenotypes that we see? So this was sort of my first foray using my now favorite method, which is single-cell profiling. And I took skins from fish at 3 weeks post-fertilization, with and without thyroid hormone. We also labeled all the neural crest derivatives in these fish using a Cree-based transgenic. And I took these skins, I dissociated the cells, subjected them to single-cell RNA sequencing to be able to basically separate them based on their molecular profiles. And just because we don't have a ton of time today, I'll just show you that I generated this post-embryonic neural crest-derived cell atlas, which of course contains the cells we really cared about, the pigment cells, but a bunch of other neural crest derivatives in the skin, which include the Schwann cells, other glia, peripheral neurons, and a ton of progenitors, which we still don't know much about. So if we zoom into the pigment cells, we had a pretty clear hypothesis going in, right? We knew these fish looked very different. We knew what cells we were looking for. And so we expected to see maybe some of those differences reflected in the molecular profiles. But that wasn't true. So this is just a zoom in of the UMAP, where each dot is a cell. And the cells are basically clustered relative to their transcriptional similarity. And you can see the general pigment cell types here. And red is without thyroid hormone, and blue is with thyroid hormone. And you can see pretty nice mixing, right? Suggesting that the molecular profiles aren't really changed so much. And the cell type proportions are kind of there as well. So this was a bit disappointing, but because we have high-resolution molecular data, whole transcriptome information under each one of these dots, we could go quite a bit deeper. And that's what I did. So if I took these cells and looked for just one lineage, so the yellow xanthophores, and looked at the genes that were changing over their differentiation time, I found that it was a small fraction of the transcriptome, but it was not nothing. So I went and looked at those genes and identified one interesting candidate, which is called SCARB1. And this candidate is a receptor that enables the incorporation of yellow carotenoids into cells. And you'll just have to believe me when I say that yellow xanthophores get their pigments from two different yellow pigment types. One of them is pteridines and the other one is carotenoids, and cells cannot make their own carotenoids, and so they have to get them from the diet. We don't feed carrots to the fish, but carrots have a lot of carotenoids, so I use this as an example here. And there was pretty recent research that had come out during my PhD that showed that if you knock this receptor out of canaries, which are the traditional yellow bird, that they actually don't have any yellow pigment. So I tested this, the role for this receptor by knocking it out using CRISPR in zebrafish, and I saw a really drastic loss of yellow pigmentation in the xanthophores. The important thing to note is that the cells themselves are still here. They just don't incorporate this yellow pigment, and that was actually what was happening in our thyroid hormone depletion fish as well. So what about the melanophores? If we do the same analysis and look at gene expression changing over developmental time or pseudotime, we saw many more genes, or a fraction of the transcriptome that were different, but it was kind of hard to pinpoint the specific things that were underlying these differences. So we went back and looked at the cells themselves. So we saw some maturation factors, and we also know that melanophores, as they become more and more mature, they often become binucleate. So this is a picture of a fluorescent melanophore in the fish skin with one nucleus, and typically about 65% of the cells at later stages of development will have two nuclei. And we found that without thyroid hormone, only about 20% of the cells were doing this, suggesting that these cells were failing to mature properly. They were getting to the place they needed to be, they were specifying, but they weren't maturing. So all of this data together and some additional molecular analyses led us to this final model to understand how this one circulating hormone was actually driving different phenotypes and two related cell types. And that was that once these cells were specifying, so we have progenitors giving rise to melanoblasts and xanthophore progenitors, that the terminal differentiation of both, which rely on different molecular and sort of morphological features, were both thyroid hormone dependent. So we're still trying to figure out, or the lab that I worked in before is still trying to figure out how this works, but using single-cell sequencing and going from the molecular to cellular to organismal phenotype allowed us to understand this process much more quickly than we would have been able to without these tools. All right, so one thing I didn't mention is that these pigment cells actually exist in a very complex skin environment, right? And another study that I did was to try and understand how this sort of post-embryonic skin gives rise to these beautiful bony scales in the zebrafish. So maybe you know that zebrafish are covered in scales, but you may not know that they develop these scales during post-embryonic development in about 5 days. So it's a super rapid process, and it happens the same basically in every single fish, and this is an amazing time-lapse movie taken by my former colleague and collaborator Andy Amon. And this is also interesting from a developmental perspective because at a single time point, which is this one here, about 4 weeks into development, you have actually scales at every single developmental stage. So this fish is one age, but you have scales at many. And that's really useful when we want to understand sort of the process of the cells giving rise to these. So at this time point, you can see we have a pre-scale, so there's no sort of, you just have the two epidermal layers, and then you have a baby scale, and then a more mature scale, and they're going through this sort of morphogenic process. And the overall goal of this study that I worked on with Andy was to resolve mechanisms of skin patterning and morphogenesis at the molecular and cellular resolution. And we used, again, single-cell RNA sequencing to profile all of the cells at this time point where we knew we would capture basically pre-beginning and late stages of scale development to try and understand this developmental process. And to give you just a little taste of what we were interested in and discovered, we have to go a little bit back in time, about 450 million years ago, to the Ordovician Sea. And from the perspective of understanding mineralized tissues, this is a very important time in evolutionary history because this is when a well-stratified ecosystem developed in the oceans, meaning that animals had to evolve armor to prevent getting eaten by other animals. And so this is really the period in time where bony skin appendages first appeared in animals like this one, which is called an ostracoderm, which literally means bone skin. And they had a body covered in these bony plates. And one of the benefits of these animals is that bone and mineralized tissue fossilizes really well. So we actually have a lot of really nice examples of these animals. I think this is a better representation. It's kind of hard to tell from the fossil. But we have really nice ultrastructure information about these bony plates, and we also know something about their mineralized composition. And the very interesting thing about these dermal appendages is that they are ultimately homologous to human teeth. All of these structures, which are sort of this bony this underlying bone covered by like a matrix that has more holes in it, like dentin, to enamel on top, is something that we see early on and is conserved all the way to human teeth. But zebrafish scales and scales of other bony fishes are really derived. So they are much more like this very flat scale. And with ultrastructural analyses and other morphological analyses, there's been a lot of debate as to how homologous these mineralized tissues actually are to those of the ancestors of these fishes. So going in with all of this sort of work and understanding of mineral biology behind us, we hypothesized that the epidermis of zebrafish retained some ancient enamel deposition function, because we do know that there's a very dense mineral matrix on the top of these fish scales. But we needed a molecular view to understand this. And so when we looked more closely at these cells, we first had to sort of orient ourselves. Here's the basal epidermal cells. We can see in these really nice in situ hybridizations that the different gene expression represents exactly what we see in the fish scale, where we have this sonic hedgehog expressing sort of outer scale layer. And when we look for specific genes that are involved in enamel production, which we know now is enamel-related proteins, we find these two key genes, ameloblastin and enamelin, in this little knob of cells that's uniquely sort of coming out of these basal epidermal cells. And if we look specifically at matrix binding dyes, we can see that right at the edge of the scale, you have this sort of ultra-high mineral content, which relates to exactly where these cells are. And we can verify that using in situ hybridization to basically further our hypothesis that this enamel matrix producing cells are still in zebrafish and homologous to the enamel matrix producing cells in other animals. So if we zoom out to understanding how this fits into the broader scale of evolution, I just pointed out how an intriguing link between molecular properties of cells match between scale and tooth formation in humans, but it's actually these same cell types that have evolved to form diverse skin appendages in mammals and other vertebrates, such as bird feathers and dog fur. And we know something about the sort of life history of these animals that might have given rise to this. So as early tetrapods, like Tiktaalik, which is a very nice fossil and visual representation, moved to land from water, they lost their heavy skinned appendages in favor of lighter, keratinized versions, such as feather and fur. And of course, I like to depict the peak of keratinized skin appendages here. But ultimately, understanding the evolutionary history and the conservation of these molecular processes, cell types, and tissues can really inform our understanding of our own skin biology. And I don't have time to get into this today, but in the study that I did with Andy, we looked at we used single-cell profiling and other genotypes of pathways and genes that are important for scale development, but are also involved in human skin pathologies to identify more molecular mechanisms. Okay, so shifting to the last topic, which is really scaling up these kinds of perturbations, single-cell scale analyses to understand organismal phenotypes, I'm just going to give you a brief overview of the method that I developed as a postdoc and now we're using in my lab quite a bit, which is called embryo skyplex. And this is really a workflow for multi-scale phenotyping of development. We go down to embryos because actually, if we want lots of numbers, looking at embryos is quite a bit more feasible. And we're really using CRISPR perturbations, which we can introduce at the single-cell stage, and embryo barcoding to basically identify differences in individual animals when we're profiling the individual cells, and a really powerful single-cell approach, which has kind of come online during the time that these stories I was telling you about, which is combinatorial indexing-based, or SCI RNA sequencing. And if you're in my lab, you hear us talk about this a lot, but it's really powerful, one, because it allows us to profile transcriptomes of millions of cells if we want to, and it's much, much cheaper than alternative methods. And I first deployed this to look at 23 different genetic perturbations and about 2.6 million cells during zebrafish development. And I'm not going to talk about the specific findings today, but I do want to highlight that in this UMAP, where we have all the different transcriptomes of the cells and how they relate to each other, with the individual embryo measurements, they can really nicely transition to looking at cellular and ultimately organismal phenotypes. So this is a plot where I'm showing you all of the different genotypes that we profiled by the developmental time, and the dots themselves are colored by how many cell types are differentially abundant. So when we gain and lose cell and tissue types, we can now quantify that by counting the cells across the individuals. So this has become really, really powerful from asking many different types of questions, not just in embryos, but also in organoids and other tissue types. And where we're really going with this in my lab is to basically scale up the kinds of questions we're asking, still to ask questions about neural crest-derived tissues, because those are kind of my favorite, but to understand differences in perturbations, and in addition to transcriptome information, understanding cellular history, so the cell lineage in an animal and how that kind of shapes the final outcome. And all of what I showed you today is really using a really powerful model system, the zebrafish, but maybe you can tell that I really appreciate an evolutionary perspective. So now that we're gaining more and more traction of these tools, we can apply them to other systems, starting with Medaka, which are here in-house with close collaborators that are also my faculty colleagues. So that's kind of where we're going in the broadest sense, and we really are still trying to answer this question of how do cellular phenotypes drive morphological diversity. If you haven't been convinced that this might be relevant societally now, I maybe will get into just a couple of final points, which is that when we understand how small changes in variation might affect model systems that we study in the lab, this ultimately sheds light on how we can interpret genetic variation that we see in humans. So one clear example of very continuous genetic variation in humans is that's displayed in this very beautiful sort of artistic Pantone display of human hair and skin pigment. And this continuous variable, which we can see from a paper published last year, is also known to be highly polygenic. So meaning that there are many different genes in our genome where small changes control sort of the continuous spectrum of this phenotype. And we know from work over sort of decades that many of the same genes and pathways that we might discover by studying zebrafish stripes or mouse skin can actually contribute to our understanding of natural diversity and disease variants in human populations. So if we think about the genetic toolkit that is being used to give rise to these different phenotypes, you may also know that these kinds of phenotypes in many in humans will also respond to environmental changes, maybe changes in sun exposure or UV exposure relative to altitude, pigment changes over aging. And so where we're going with this genetic understanding is to try and interpret how these changes also are impacted by the environment, which is a really tricky question. So I'm going to let Gilles tell you more about the environmental impacts on development and organismal physiology. And I will wrap up there by thanking the people that I have worked with over my career, my current lab members who I really, really enjoy working with every day, Andy Amon and Sandri Suvatsan, who were really close collaborators of mine over the years, and my growing network of collaborators in my current scientific home, Heidelberg, and of course the fish over all the years. And I'm very happy to take any questions.
Thank you so much. Thank you, Lauren. It was a fantastic view on your research and the relevance. We really liked the last slide. Good. We're going to move on with the next speaker, Gilles Storelli. So while he mic up, I'm giving you a few words about him. So Gilles is studying the gut microbiome, and he uses Drosophila as a model for this. And he's in particular looking at how the gut bacteria interact with the metabolism of the host and interfere or benefit or contribute to its nutritional profile. So he's based here at COS, and he joined us in 2025. He's originally from France, and word of disclosure, this place, COS, is going to go down the drain because we reached a critical mass of 2 French people among the faculty. We warned you. And he studied development, genetics, and immunology. He did that in Marseille. He earned his PhD in Lyon, focusing on already host microbial community interaction already in flies. And his PhD is secured in 2015, and he then moved to Utah for a postdoc. There he uncovered the lipids that are stored in the embryo are converted into protective compound when the embryo fly embryos morphs into an adult. And that helps to, so the molecular mechanisms responsible for this transition in profile of usage of lipids. So in 2020, he came back to Europe and became a group leader in Cologne, advancing his research on how the microbiome shapes the physiology of the host. And his work, and during that time, he earned a couple of very prestigious grants and major funding, including an ERC starting grant 2 years ago. And since the beginning of the year, so 2025, he has been here a professor for molecular organismal biology at COS. And welcome, Gilles. We're very happy to have you today.
Thank you, Alexis, for the introduction. So everyone can hear me? Yeah, I guess it works. Okay, good. So I guess that you are all aware that trillions of bacteria live in our gut, and they shape almost every aspect of our lives. Therefore, a major objective of current microbiome research is to understand how shifts in these populations can basically provoke disease. But this quest faces major challenges because there are very basic factors or parameters of host microbiome interactions that we still do not understand. For example, bacteria can produce metabolites or molecules that will affect the physiology or the function of tissues. But in fact, these molecules can act in a very indirect manner to exert these effects. And for most of them, we cannot really predict their mode of action. In parallel, one aspect of host microbiome interactions that is neglected is how the host itself, or how the intestine shapes bacterial physiology and bacterial metabolism. Because you might expect that a same bacterium, depending on where it is in the gastrointestinal tract, can produce completely different metabolites, and this is something we don't really understand today. So this is why I'm convinced that to understand the outcome of host microbiome interactions, we need really to integrate studies in animals and in their gut bacteria together in order to understand how the two partners cooperate basically to support health and metabolic homeostasis. And so these are, I feel, the most important challenges that microbiome research faces today. And we, with all of our colleagues, are trying to basically answer those questions. And in particular, in our lab, we study these interactions from 3 different perspectives. So we are really interested into the bacteria themselves, and we want to understand how they adjust their metabolism depending on where they are in the intestinal tract. In parallel, we are naturally interested into knowing how bacterial factors and molecules will then impact host physiology and in particular the function of the intestine. And finally, we want to understand how the 2 partners cooperate for the greater good. And in other words, how do they complement each other to fulfill their metabolic and nutritional needs. And these research objectives are ambitious, but they would benefit from a simple symbiosis model that allows manipulation in the animal, but also in its gut bacteria. And the fruit fly, Drosophila, fill all of these criteria. And they do so because first we have a wealth of genetic tools in this model that allows to perform genetic manipulation in specific cell type and in a time-controlled manner. But in addition, the fly digestive system is similar in terms of anatomy, physiology, and function to that of mammals. And this is what I try to highlight here with these different colors. As you can see, or barely see with my laser, I guess, but you have an organ that is analogous to the stomach in flies, then you have a portion of the digestive tract that is similar to the small intestine, and then we have a portion that is equivalent to the large intestine in fruit flies. So flies have a gut, and obviously they have a microbiome. And the good thing here is that the microbiome of flies is relatively simple, so maximum 100 bacterial species, some of which are also found in the microbiome of vertebrates and even mammals. Another advantage of the fly here is that most of the bacteria that are found in their guts are also easy to culture in the lab. And finally, it's relatively simple to maintain flies in germ-free conditions, so in the total absence of microbes. And this is a very important parameter because then we can reassociate germ-free animals with microbes of interest to basically understand how these specific bacteria can shape host physiology. And finally, both flies and their gut bacteria are compatible with genome-wide studies, and this makes them one of the most powerful symbiosis models to discover the mechanisms that regulate host microbiome interactions before the validation of those mechanisms in more complex organisms such as mammals. And I will give you an example in the next few slides. So today, fruit flies are well accepted as a model to study host microbiome interactions, but it took a bit of time to get there. And in particular, when we started using this model to study these interactions with François Lellier in Lyon, in France, one of the first questions we had was, do gut bacteria indeed influence nutrition in fruit flies? And to answer this question, we looked at one specific parameter that is highly dependent on nutrition. And this parameter is developmental time. And so basically, developmental time here is the time taken for the embryo to reach the adult stage, and it's highly dependent on the quality of nutrition of the fruit flies. So put it simply, if you feed flies a rich diet, they will develop fast. If you feed them a diet where you deplete specific nutrients, they will develop slower. And this is what you can see here. So here, what is depicted is the timing of adult emergence of germ-free flies, so flies without a microbiome, when they are fed a rich food or when they are fed a food in which we reduce protein content. And as you can observe here, and in agreement with what I just told you, basically it takes more time for adults to emerge when flies are fed a diet that is deprived of protein. So given that this parameter is highly dependent on nutrition, is it affected by the microbiome? So we've performed similar analysis this time on animals that carry a microbiome. And on a rich food, there is barely no difference. So if animals have a microbiome or not, they develop at exactly the same pace. Now, when we've done similar experiments on a poor protein diet, what you could see now is that animals with a microbiome, they develop much faster. And this shows that gut bacteria protect flies against the deleterious effect of malnutrition. So the next step for us then was to determine or to identify the bacteria that are responsible for these effects. So what we've done, we identified basically bacteria that were found in the microbiome of those flies. Then we isolated bacterial strains, and then we reassociated germ-free flies with single bacterial species, and we scored development on this poor protein diet. And to cut a long story short, what we could see is that reassociation with a single bacterial species could recapitulate the effect of a complex microbiome. And this bacterial species is Lactobacillus plantarum. So it's a lactobacilli, and it's a member of lactic acid bacteria. And basically here, what you can see, so this is a picture of larvae that were reared on a poor protein diet. And these larvae have exactly the same age. The only difference is that on the left, they are germ-free. On the right, they have been associated with Lactobacillus plantarum. And you can see the massive growth gain when they're associated with this particular bacterial species, which then translate into faster adult emergence on a poor protein diet. And I want to stress the fact here that the strain that we have used for the inoculation has been isolated from the gut of flies. And we wanted to know whether all Lactobacillus plantarum isolates, wherever they would come from, would have the same effect. So we tested inoculation with other bacteria from the same species, but we could see that this effect is highly strain dependent. Because here, for example, this is the effect of a strain of Lactobacillus plantarum that was isolated from a food product. And you can clearly see that it has an intermediate effect on growth, but it has not as a growth promoting as the bacteria that was isolated directly from the fly gut. And what this shows basically is the complexity of host microbiome interactions, because some bacterial effects are not even shared within the species. They can be strain dependent. In addition, one question we had is, given that Lactobacillus plantarum is found in the gut of flies, but it's also a natural commensal in rodents and in humans, we wanted to know whether it could have similar effects on mammals. So for this, we had collaborators who did the following experiment. So they had germ-free mice that they weaned on a poor protein diet. And these mice were either germ-free, and they score growth of these animals, and as you can see for the germ-free mice, when they are weaned on the poor protein diet, basically their growth is completely stunted. They grow better when animals have a microbiome, which recapitulates what we've seen before with flies. And what you can observe now is that when germ-free animals are reassociated with the lactobacillus that were isolated from fly, these animals, they grow as well as if they had a diverse microbiome. While now the strain that has an intermediate effect in flies also has an intermediate effect in mice. So altogether, this really highlights the need of simple symbiosis models such as fruit flies or insects associated with single bacterial strains to discover complex bacterial effects on physiology. So then the next question you might have is, how do bacteria exert those effects? How do they complement the nutritional needs of these animals? Well, bacteria could fulfill these nutritional needs by different manners. So they could act directly, because bacteria are well known for producing nutrients that animals cannot produce, such as essential amino acids or vitamins. They can also maximize nutrient extraction from the diet, because bacteria can digest nutrients that animals cannot break down. But what bacteria can also do eventually is have an indirect effect and alter the function of the digestive tract itself so that it becomes more efficient at assimilating nutrients. And in the case of flies, all of our data points toward this indirect regulation of intestinal function. And for now, this is the model that we currently have for those effects. So in all intestinal epithelium, from insects to mammals, the cells express specific receptors that are called pattern recognition receptors. And these pattern recognition receptors, they are in charge of sensing bacterial cues. And here, in that case, what they recognize are fragments of the cell wall of bacteria that are released when they divide. And the major function of these pattern recognition receptors is to, after recognition of these bacterial cues, to induce an immune response and induce the production of antimicrobial peptides or antimicrobial molecules that will then kill bacteria. And this basically allows the maintenance of a steady state of population of bacteria and to limit infection. What we've discovered, however, is that these pattern recognition receptors also act via a completely independent pathway to remodel intestinal metabolism. And in particular, what they do is that they suppress carbohydrate and lipid digestion while they increase protein digestion in enterocytes of the Drosophila epithelium. And our latest data suggests that this metabolic remodeling is another way for the intestine to control luminal populations, because some bacterial pathogens and bacterial parasites rely on carbohydrates and lipids that are digested by host activities. So we think by suppressing these activities here, this allows the control of this potentially deleterious bacterial population. But at the same time, this induction of protein digestion will become beneficial now when animals are put on a poor protein diet. So basically, this effect on growth, we think, is a side consequence, basically, of this metabolic remodeling of the intestine. And altogether, this shows also how nutrients are really central, basically, in dictating the outcome of host microbiome interactions, because they are highly nutrient dependent. And for the few minutes that I still have left, I would like to give you another example of how nutrients are really central, basically, in shaping this interaction between animal host and their gut bacteria, or basically the outcomes of these interactions. And this second part basically is still unpublished, but it started with this observation that we had that when we were associating flies with different gut bacteria, including the lactobacilli that I just talked about, we made this interesting observation that most of the time these animals would show retention of food in their crop, which is the equivalent of the stomach in flies. So this is what you can observe here. So what you have here are a few crops or stomachs that were dissected from flies that were inoculated with PBS, so it's just a vehicle control. And here you have the same picture for animals that were inoculated with lactobacilli. So flies inoculated with lactobacilli, they show this food retention in the stomach. And when we look at higher magnification into the content of the stomachs, we see that there is more food, but also the food has a different structure. Because in animals that are inoculated with PBS, what we could see is that the food is present in the lumen as single particles that are freely floating, while now when they are associated with or inoculated with lactobacilli, food forms these very large and very dense aggregates. And when we zoomed into these aggregates, what you could see is that they indeed contain food particles, but they also contain a lot of bacteria, as you can appreciate here. So here, what happens is that bacteria will recognize food particles, they will bind to these food particles, they will then bind to each other, and this allows them to maintain together food particles that are 100 times bigger than themselves. And following this observation, we made the following model. What we think happens is that the fly will ingest food, then the food will enter into the crop or the stomach. This is where the mechanical processing of food will start, and this is also where bacteria will start getting in contact with these food particles. Then bacteria will bind to these food particles, they will bind to each other. This will lead to the formation of these large food agglomerate in the lumen of the crop. And this is the reason why we think then food is retained longer in the stomach, because basically these agglomerates are too large and difficult to break, and they are too large to pass through the stalk, which is basically the connection between the crop and the small intestine of flies. So the next step for us was then to determine what do bacteria recognize on food, what do they stick to. So we looked at the nutrients that were present in our experimental diet, and what you could see is that the diet we give to our flies is highly rich in dietary fibers. And dietary fibers, basically what they are long polymer of carbohydrates that cannot be broken down by animal digestive activities. For example, we could think about cellulose or hemicellulose. And what we know on the side of bacteria is that they express adenosines that can recognize some of these carbohydrate moieties that are found in those dietary fibers. So we wondered whether the recognition of fibers by bacteria was responsible for the formation of these food agglomerates. So what we've done to do that, we mutagenized these adenosines, and then we tested the effect on flies. And this is what we could see. So on the left, so this is a video of a stomach of a fly that has been inoculated with the wild-type bacteria. As I could show you before, food, which is labeled here with a fluorescent dye, will form a very dense, packed aggregate in the stomach that is hard to move out. While now, when flies are inoculated with the bacteria that is mutant for the adenosine that allow them to recognize and bind to these dietary fibers, now food is evenly distributed in the stomach and does not form these agglomerates anymore. Yeah? Then we zoomed onto the stalk, which is the connection here between the stomach and the small intestine of flies. And you can see that these large aggregates with wild-type bacteria are particularly hard to pass through this narrow section of the digestive system, while lactobacilli that are mutant for these adenosines that allow them to recognize fibers, in that case, food particles remain isolated and they pass freely through this narrow sphincter of the digestive system. And altogether, what this shows basically is that bacteria can regulate transit by altering the physical structure of the food. And what I could show you is that this effect relies on the bacterial adhesion to specific nutrients, and in that case, to fibers that are present in the food that we give to our flies. Another process that we think is really key for the formation of these large agglomerates is bacterial autoaggregation, so the fact that bacteria bind to each other. Yeah? And bacterial autoaggregation is a complex behavior that is poorly understood. We think it's involved in transit. It might also be important for bacterial survival and persistence in the digestive system. So we are particularly interested today in understanding how this bacterial behavior is regulated in vivo in the gastrointestinal tract. So for that, we started a genetic screen where basically we use a collection of single gene knockout in our bacteria, and then we screen them for altered aggregation phenotype. And we already found mutants or strains that show loss of function phenotype where basically bacteria cannot bind to each other anymore. But we also have gain of function phenotypes where bacteria show an aberrant aggregation with each other. And we hope that this screen will allow us to identify the pathways that regulate this behavior in vivo in the digestive system. And finally, one evident follow-up of those studies is to understand what is the impact on nutrition in both flies and their bacteria. Because here you might think that this process is beneficial for bacteria because they are really close from their food source, so it's very likely they can maximize nutrient extraction by being stuck to these food particles. So this is something we are currently testing. And for the fly, it's not clear yet what could be an impact because it could be both positive and negative. Because here we might imagine that because bacteria are really all around the food particles, that eventually they might compete for nutrient availability with flies. But at the same time, given that it takes more time for the food to transit through the digestive system, maybe digestion is enhanced, and at the end, animals could extract more nutrients. So this is two scenarios that we are currently testing. So to finish, and before leaving you, I would like to give you again these few take-home messages. So bacterial effects on animal physiology are complex. And as I could show you today, for example, a single bacterium can have different effects. So in the case of lactobacilli, they can induce protein digestion in the gut, but at the same time change the structure of the food, which could have then dramatic impact on nutrition. And in parallel, these effects are strain-specific. So this means that different isolates from the same species could have completely different impact on physiology. In addition, these effects are localized. So I told you about this food retention in the digestive system that was really driven by the anatomy of the crop and the fact that we have this sphincter between the stomach and the small intestine. What we are also interested in is how the luminal environment also shape bacterial physiology and metabolism, because we might expect that the luminal environment change, for example, along the small intestine, and therefore bacteria could also adapt to these changes in their environment. And finally, these effects are highly nutrient dependent. I could show you that, for example, the outcome of host microbiome interactions can be seen in specific nutritional conditions, such as a poor protein diet. Then there is this interaction with fiber and the formation of these food agglomerates. And we are also interested into xenobiotics, and in particular preservatives that are in food and that might impact specific bacterial function. And all of these really highlight the need of simple symbiosis models such as invertebrates associated with single bacterial strains to really understand how bacteria shape animal physiology. And with this, I would like to thank the people who were involved in the work, especially the unpublished work that I present in the second half of the talk. So Karin Fiedler, Aparna Kelkar, and Fenja Meyer-Zwell-Tenschildesche were parts of our lab in CICAD in Cologne. And as you have all heard, we've recently moved to COS 3 months ago, and we are hiring. So if you're interested in our models and our questions, please feel free to contact me. I would like to thank the funding. I would like to thank you all for your attention, and I will be answering your questions later. Thank you.
Thanks a lot, Gilles. This was absolutely fascinating and definitely make us consider differently the other holes, I mean, the millions and millions of bacteria we have in us. So please, Lorraine and Gilles, come back on stage. Now it's the moment where you can talk. So sit here and you can use that mic. So I'd be happy to take questions. Marcus is the first one. Marcus is a local, so he knows what this thing works. We are testing several skills here.
Oh. Perfect. First of all, thank you to both of you. These were two perfect talks. I really enjoyed them. I have a question for Gilles. When you say you feed these the flies with the food, is this an infinite access to food? So do they have enough, or is this limited? Does maybe if the bacteria would influence their digestion behavior or their feeding behavior, would that also make a difference?
So the flies are fed ad libitum. So the way we maintain them is that they are put into plastic vials where they have continuous access to nutrients. But what we've observed is that if you refer to the condition where we have this retention of food in the stomach, what we see is that there is a slight increase in food intake in these animals, maybe to compensate for the slower transit. So this is what we observe. But at the same time, given that food is retained longer in the stomach, there is also a dramatic reduction in defecation rate. So they feel a bit more, defecate less, because it takes more time for the food to transit through the digestive system.
Okay, I think Matthias was a bit faster.
Okay, I also have a question for Gilles. This was really very nice. So first of all, the agglomerates, are these biofilms in the classical sense, or do these bacteria secrete polysaccharides and whatever proteins, DNA, form this?
Yeah, so there is already direct physical binding, as we could see, but yes, there is very likely production of biofilms because we use different stains, and we indeed see that there are exopolysaccharides that are produced on top of food. And this might also have a further impact on then how the food is digested, yes. Yeah.
And do they also produce exoproteases to digest the proteins?
Yes, they do. Yeah. So yeah, they secrete actively proteases that allow them to degrade proteins that are in their environment. Yeah.
Then of course it would be interesting to know what the specificity, the cleavage specificity of these proteases is as compared to the proteases that is used by the flies themselves.
Yeah, so this we don't know, but it's very likely anyway that the proteases are able to digest the food that we give to our flies, because the bacteria can survive on this already, but we don't know basically if there is a specificity to the proteins that are produced, but we know for sure that they allow bacteria to metabolize this food. Yeah. And whether they are in competition with flies or whether bacteria digest partly protein that can be used by the fly, these are things we are currently testing right now. Yes.
Thanks a lot.
Right. Jan?
Right, how does nutrition affect the developmental speed of zebrafish? I guess in the early stages, not at all, because they feed on the yolk, but then later on?
This is a question for me. Okay. Oh, really testing the limit of my knowledge. So what I do know, which is maybe not the most up-to-date microbiome research in zebrafish, is from Karen Gilliman's lab in Oregon, where if you raise fish in a germ-free environment, certain cell types in the gut will not differentiate properly. So they have issues in sort of early morphogenesis if you do not have the early gut microbiome.
I was just wondering about nutrition. So
just nutrition.
Exactly. Did you feed them on different types of food, or you give more or less? How would that affect the speed, and at what developmental stage does this sit in?
The zebrafish feed on the yolk until about 4 or 5 days post-fertilization. So until that point, it's really controlled. And the yolk of zebrafish is different than the yolk of many other fishes, where it's not very lipid. It doesn't have very many lipids in it. It's much more protein dense. But manipulating the yolk amounts is kind of difficult because it's these sort of granules that are stuck inside this balloon, more or less. But there are labs that work on the transition of nutrients from the yolk to the early embryo. The connection to developmental speed is not one that I know that well, but if you remove nutrition, then the fish don't develop as well, which can be observed probably as a developmental delay. But good question.
Gaspar, then on the front.
To Laurent, also about growth in fish. So these thyroid-ablated fish, do they later have the growth phenotype?
Yeah, they develop goiters, just like human patients with thyroid disorders do. But, you know, studying thyroid hormone disorders is not necessarily something that's really of interest to the medical community because it's very easy to treat thyroid hormone disorders. You can just give people thyroid hormone for their whole life and they're fine. But the gross morphological differences are much beyond pigment. They have very thin skin, they don't grow as fast, they die early, they can't reproduce very well, they have these goiters, and there's other neurological defects too that we don't really understand very well.
There is a question front row first.
I have a bit fundamental question on that. Why is fish, or why zebrafish in particular?
Why do we study zebrafish? Okay. I can start with a broad answer, which is that we are all fish. But I can get more specific, which is to say that if we look at the sort of the genomic similarities between zebrafish and humans, for instance, we find that are about 60 to 70% of protein coding genes that are homologous between zebrafish and humans. And we know from decades of studying how these individual genes in the zebrafish genome impact development of specific cell types and specific organs, that those often translate very well to looking at sort of in the clinic and seeing differences and developmental disorders that arise in human babies, and have really enabled us to identify specific mechanisms that help us understand disease processes, but also normal development. So that's sort of a very broad answer. But then from a practicalist perspective, zebrafish are really fantastic to study the development of because they develop externally from mom. So you can set up your fish in the evening, collect embryos in the morning, you have hundreds of them, and then you can, you know, take them and look at them and observe different cellular processes and molecular differences. So there are practical and also fundamental biological reasons that we study them.
I was afraid that you would say that they are better than flies. You know, it's okay.
No. We need to understand all the different organisms, Gilles.
So maybe along that line, before I gave the mic to Casper, so both of you are a wonderful example of how by studying model organisms, you can directly connect to things like with more, you know, health related or with us human. But what are the limitations in there? You know, is it really perfect model, or what should we care or pay attention to?
No, I mean, they're obviously not perfect model because we cannot really address I mean, if I take my field, for example, we cannot really fully address the complexity that you would find in a more complex organism and with a more complex bacterial communities. But at the same time, this is the only way for us to find those basic mechanisms and eventually then influence them in a diverse bacterial communities. This allows us to have intervention means, basically, to try to shape these communities to make them do what we want them to do. So they are not perfect, they are limited, but at the same time, I think they are necessary to just describe these very basic principles.
Agree. And the more that we understand the individual models we work with, the more we understand the limitations and the similarities. You know, if I want to understand mammalian bladders, I'm not going to look to a fish, I'm going to look to a dog, right? Because there's specific models that are very good. And so when you have the questions that you're interested in, you can kind of look to the model that's most relevant and also bridge across different aspects of biology to really understand.
Yeah, I think it's a very important point.
I have one question for Dr. Storelli. Is in one of those slides there was a mention of if we have a microbiome or gut bacteria, then the fly development increases even though it's a malnutrition. Applying that in terms of societal or in development of humans, can there be, or has there any thought given to develop a superfood or a medicine for people who are living in malnutrition countries and so on?
No, for sure. I mean, this is one of the follow-up of these studies, and that this is followed basically by my previous mentor, where I had done this preliminary work, and this is what they are aiming for, basically to give those bacteria as potential probiotics or supplementing, fortifying food with those probiotics to enhance nutrient assimilation in malnourished children. So that's definitely what they are aiming for now. And this is why this step of validation in mammals was really key then to have the next step, prepared for eventually these clinical trials in humans. Yeah.
Super. Thank you.
If you can throw the cube. We're well coordinated here. Andres, yes, perfect. Casper.
Thank you both for those fantastic talks. And I have a question for Lauren. So you, of course, are heavily now involved in single-cell genomics, as you mentioned, and of course, also that you mentioned that integration is really important. I completely agree that. But then, what do you think, or what do you want to be like the genomics technique of the future in case of looking at single-cell resolution, or something completely new? What would you like to work on in the future in that regard?
Yeah, thanks for that question. Well, I think in terms of single-cell omics space, things are moving really, really rapidly, right? And the natural thing is to start with one modality, or RNA, for instance, and then just layer and layer and layer, because if you think of like the most complex picture, what you have is single-cell resolution, you have RNA, protein, spatial information, developmental time in 3D. It's literally recreating sort of the molecular and cellular atlas of an organism through time and predicting it, right? So we're going to need AI to be able to do that. But then in terms of what I really want to do in my lab to build on these comes down to the questions that we're really interested in, and that is understanding, right now we're taking RNA, which is the best way right now to capture what a cell is sort of thinking, I like to say, the fate of the cell in the moment, and sort of connecting that back to its developmental history. And we do that at a snapshot where we can't see what the cell has done in the past. So that's why we're really pushing on building in lineage information, so that we can take a cell, look at its transcriptome, and also see who it's related to in a sort of general vicinity. And then we can really ask questions about, if you remove a certain population of cells, has another one filled in where we couldn't have measured that before? And how do these kinds of things change if you change the environment or the genetics? So I think it comes down to the question, but of course, would we like to have spatial information there? Yes. Would we like to have proteomes? Yes. And so I think just more, but more is not always better, so that's where the specific question, I think, is really important here too.
Oh, okay, thanks.
I see that a significant part of the audience is way below 30, and we haven't heard any of those voices yet, so I really encourage the students in the audience to ask any question they have. How did they become scientists? Oh, please, one.
not a student anymore, but I still have a question. Thank you both for the exciting talks. So I have a question for Lauren. I was wondering about the thyroid hormone and I understood that this affects the uptake of carotenoids, but I didn't quite get how. So does it basically regulate the expression of SCAR-B? And if so, how does this work exactly? Can you elaborate?
Yes, thanks for the answer. And I have to start out by saying that we don't 100% know for sure. But as I mentioned, the receptors for the thyroid hormone is actually also a transcription factor. So these receptors will bind the hormone as it comes in and very likely turn on or turn off specific genes. And SCAR-B run is a hypothesized sort of direct regulator, or a gene that's directly regulated by these receptors. These receptors also do something even more complex. So when there's no thyroid hormone there, they're not just sort of free floating, they're actually bound to DNA and blocking transcription of certain genes. So another thing I did in the study was to sort of remove the receptors and also remove thyroid hormone, and we saw that a lot of the phenotypes were actually rescued. And so it's the repression that acts by the receptors that also contributes to these phenotypes. So understanding exactly how that works is not totally understood yet because they're binding all over the genome, but it's definitely a question. That's a really nice follow-up to what I showed. Thanks.
Thank you.
Question at the very back.
Thanks for the great talk. This would be a question to Lauren, and it's more a technical question. I was thinking about the fact that melanophores getting binuclear when they mature. Is it easy to capture this with the single cell analysis, or is it also different if you would do nuclear sequencing? So can you see this in your data set?
Yeah, that's something, Bettina, that we were really hoping to see. So the pigment data set that I showed you is actually single cell, and then after that, the methods I used were single nucleus. So in single nucleus, we would have dissociated the cells and never been able to put them together. But we were really hoping to be able to see this, and the way that we looked was looking for maybe almost double in the amount of genes expressed or something like this, but we really didn't identify this, and one reason might be that we don't know the sort of transcriptional activity of these nuclei, and it could be that one sort of gets more silenced while the other one, there's so many questions about these binucleate melanophores, and so that was a hope of that data, but I can't say that we really identified those, so maybe something for the future. Yeah, thanks.
Bettina in the back. Zenia, and then behind you, Zenia. Yep.
Thank you. Also a question pertaining to the thyroid hormone. Is that one usually looks at like ablation, and then that you could rescue some certain phenotypes by also getting rid of the receptors. But if I recall correctly, the receptor occupancy isn't that high with thyroid hormones, so I've been wondering whether one would observe a different phenotype, like a distinctively different phenotype with high thyroid states. Also, perhaps altered differentiation patterns. Generally more activity of genes that usually are somewhat thyroid responsive, but may in certain contexts also be compensated for, like in case of ablation.
Yeah, thanks. So to summarize the question, maybe to what happens to these phenotypes if you actually just increase thyroid hormone, and if that's sort of a direct opposite of the ablation, and that is actually the case. So I didn't show any of these experiments, but if you put thyroid hormone in the water, you can create basically a hyperthyroid state, and we also have a genetic mutant in zebrafish that's a hyperactive receptor that basically contributes to overproduction of thyroid hormone that matches a variant that we see in humans actually, and when we do that, or when we look at that fish, the fish is actually really, really yellow. So it has way more sort of carotenoids in the xanthophores, and it has fewer melanophores, and basically you see sort of these differences in the two cell lineages that are kind of opposite each other. There's also other stuff going on that we don't totally understand, but that's a really nice tool that we can also use to make sure that we're not sort of biased in the things that we see with just the ablation model. Yeah, thanks for your question.
So there's
I have a couple questions. So I have actually two for Lauren and one for Gilles. I'm going to start with Lauren. So you mentioned that you like to study how molecular changes are connected to morphological outcomes through an evolutional lens. And you said that you're currently focusing on Medaka, but I'm wondering, when you're kind of at a nice point with that organism, what would you which organism would you like to go next and why?
Thanks for that question, Xenia. Okay, sometimes I dream about the organisms that I could single cell, but I can't get my hands on. Right, so Medaka, it's 150 million years about diverged from zebrafish, so still a lot of similarities, but quite far. But one of the things that we struggle with when we study bony fish is that they've gone through this extra genome duplication that, for instance, the lineage that led to us didn't go through. So it can make some of these interpretations difficult. So I would move to a more ancestral state, so living animals that sort of represent or are relatives of a more pre-duplicated state, and that would be something like sharks. Cat sharks are a really nice model.
Sharks at Kos. I'm so for it.
That sounds really cool.
The cat sharks are very sweet. And they're not very big. Or like baby skates. So they're this little skate model that are also chondrichthyes, and they can tell us. Now my lab's really interested in cartilage development. So some of these cartilaginous fishes are ones that are really interesting. And we are now getting more and more models online to sort of bridge this sort of pre-duplication to mammals. But I'm very fond of the fishes, so I would at least start there before branching out to furry things, which scare me a little bit.
Thank you for your answer. And I also wanted to say that your talk was really engaging. And the second question I have to you is, you said that transcriptomics kind of tell you what a cell is thinking, but epigenetics really influence the mRNA profile of a cell. So are you currently looking into approaches to do single cell epigenomics?
Yeah, so I didn't talk about that at all, but that's a good question. Of course, there are many different genomic features that influence gene expression, and these can be linked to what a cell has gone through in the past. So using new methods like single-cell attack-seq, which profile open chromatin, can give us pictures into what regions of the chromatin might be available to gene expression. And then we're also thinking about building tools to do single-cell cut and tag, which can look at the specific location of proteins that might open or close the DNA that lead us to enhancer regions, which will then help us understand the networks of gene expression. So maybe not just what's expressed, but what's turning it on. And these are the kinds of genomic elements that really rapidly change over evolution. So to really understand how these processes work over time, those are going to be really important things to study. So yes, we are also thinking about those and getting them going in the lab. Thanks.
Thank you so much for your answers. And the question I have to Gilles is that you talked about how the presence of the bacteria from the fly strain influence developmental time. And what I'm wondering about is, have you looked into it, or your colleagues maybe, how that affects the aging process of the fly?
So I think there was one study where they used the exact same strain to check whether you would have an effect on longevity, and the answer was there was nothing really visible in terms of adult longevity. So it's not because animals are developing faster or slower than that they will live longer or shorter. So there was no connection like this. Yeah. So we could not see anything. Yeah.
Thank you so much.
So there is a question at the very front here, which by I think the youngest member of this audience. First row. Gravity will help you. Perfect. Almost. Yeah.
Thank you for your talk. Actually have a question to both of you. It's a very broad question. Is there any application of quantum biology in any of your researches?
You didn't see that one coming.
Yeah. I mean, what would you define as quantum biology?
Anything to do with like cells or the changing of the DNA or anything like that in your research?
So more to do with things like something happening at subatomic particle level, which can alter the actual genetical level. Something that happens also in the ammonia molecule, where the nitrogen jumps between the three hydrogen, where what they call it is quantum tunneling.
So, getting really, really deep down and looking what's happening, even subatomically. Okay. The short answer is yes, that all matters. The broad answer is that the tools that we use to study the kinds of biology in our labs don't give us information about how those things are changing. And so even though they're probably very important, we're limited by what we can measure, and so we aren't detecting them. So hopefully the generation of the future will be able to start to bridge some of these really, really tiny things that can, we're going even lower than the scale that I started with DNA, right? I think that's going to be really important to sort of extend out both of those areas of scale. And so we need future biologists and physicists and chemists to really help us bridge those things. So we don't maybe have a good answer now, but maybe in the future.
I would have a question for both of you, and maybe this would be of interest to you or others here. What brought you to biology in the first place? As a student, when and what was there any decisive moment that tell you, okay, I'm going to do biology and study that?
I was, a young girl in the prairie state of Illinois, and I liked visiting the ocean more than anything because of all the animals that were there. So like many biologists, I wanted to be a marine biologist, and so I started going down the path of, understanding animal ecology and things like this. And then in college, I had my first experience in a molecular biology lab, and the ability to sort of ask questions and design experiments and then look under a microscope, I was just totally hooked. So I was interested in biology, but I didn't really know what was possible until I had these new experiences, because my family members aren't scientists. And so it was all a bit later, but that was it.
And for you, Gilles?
For me, I mean, as I was a kid, I was always lifting rocks to see what is under. And I got stung with a lot of different things. So I think I just had a natural interest for this. And I must say also for insects. So this is why we keep studying them, I must admit. So this is basically what draws me naturally to this courses. Yeah.
And again, maybe to both of you, what do you think are, for the students here, good qualities, or what does it take to be a good scientist or a good biologist?
I think the important thing about working in this field is that there are many, many, many different types of people, different personalities, different motivations that kind of all come together and work on related questions. So I think sort of the diverse perspectives are what make working in a lab, working at costs, like the absolute best thing that I could be doing with my life. But there are a few, I think, main features. You have to be curious. And if you're not curious and you don't necessarily want to keep getting to an answer, I think it's going to be really hard to get motivated through the difficult experiments, the uninterpretable data, the things that should work but really just don't work over and over again. And having a community of people around you who you can share that with, I think that's sort of the fundamental thing for me anyway.
I think it's difficult to give a definition of good scientist, because I think it's really dependent on the environment. So I think you could be a very bad scientist in a given lab or in a given environment, while you would be a stellar scientist in another environment. So what I would maybe rephrase it a bit differently. What I would encourage you young students is to find really the environment where you would thrive. And this means selecting your colleagues, selecting your mentors, selecting people that you have around you in the lab, because this is this homeostasis that makes that makes you basically a good scientist, I would say. Then maybe one thing I would add is, yeah, I think hard work is required. You have to have luck. There is a big luck factor, I would say, in this field, and you have to have hard work, because as Lauren was saying, I mean, we all face experiments that are not working. I mean, it's not that they are not working, it's that you test hypotheses, and this is probably not the good one, so you test the next one, and then the next one, and then the next one, until you find the one that is working. So, and this requires tenacity or luck, because you could find the right answer from the start, or it will take you 10 trials before you will find what you need. Yeah.
Tenacity is a good one. We need that.
I think on this wise word, we could conclude, and I would like to thank you, both of you. Really, you did a terrific job. Thank you very much.
Thank you.
I also would like to thank you for coming tonight, especially the students. I'm very, very happy that so many students came tonight. As you know, there is the opportunity to keep discussing among yourself or with Gilles and Lauren. Join us outside of the auditorium for a little things to hit and to drink. And thanks again. Welcome. Bye-bye, sorry. And see you next time.